Sickle cell disease, also called sickle cell anemia, is a genetic disease where red blood cells can take the shape of a crescent or sickle. That change allows the red blood cell to be more easily destroyed, causing anemia.
Sickle cell disease is caused by defective hemoglobin, which is oxygen-carrying protein in red blood cells. Hemoglobin A, or HbA, is made up of 2 alpha-globin and 2 beta-globin peptide chains. The beta-globin chain ends up misshapen, caused by a mutation in the beta-globin gene, or HBB gene.
Sickle cell is an autosomal recessive disease, so a mutation in both copies of the beta-globin gene is needed to get the disease. If a person has just one copy of the mutation and one normal HBB gene, then they are sickle cell carriers, also known as the sickle trait.
The sickle cell mutation is a non-conservative missense mutation that results in the 6th amino acid of beta-globin being valine instead of glutamic acid. A non-conservative substitution means that the new amino acid, valine, which is hydrophobic, has different properties than the one it replaced, glutamic acid, which is hydrophilic.
A hemoglobin tetramer with 2 alpha-globins and 2 mutated beta-globin proteins is called a sickled hemoglobin, or HbS. When HbS is deoxygenated, it changes shape, which allows it to form long polymers that distort the red blood cells into a crescent shape. This process is called sickling.
Repeated sickling of red blood cells damages their cell membranes and promotes premature destruction. This destruction of red blood cells not only leads to anemia, but also means a lot of hemoglobin spilling out. Over time, sickled red blood cells clog and cause vaso-occlusion and pain crises in bones.
The factors that cause red blood cells to sickle, which are hypoxia, dehydration, and acidosis, can all be improved with oxygen and fluids. In addition, opioids are usually used to manage pain, and antibiotics are used to treat any underlying bacterial infections. Children who are tested for sickle cell anemia during a screening and are found positive for sickle cell anemia are treated with penicillin and a polysaccharide vaccine to help prevent sepsis and meningitis.
One of the many questions surrounding sickle cell anemia and those affected by it is there a cure? In previous years, scientists deemed sickle cell anemia incurable, but recent research argues that there might be a way to cure sickle cell anemia.
These researchers believe gene therapy is the answer. In this strategy, the patient's hematopoietic stem cells will be removed and then replace the HBB gene with a healthy copy, and then transplant it back into the patient. The healthy copy of the gene is delivered to the cells using a modified harmless virus. These genetically corrected cells will then repopulate the bone marrow and reproduce healthy rather than sickled red blood cells.